Excellent. So, Vanya will wait for your clearance.
Just a minute, it's starting. Just the internet is okay, but it's going. Just a minute, Adilson. Could you please stop sharing because I need to start first. Please just.
Oh, okay.
Okay, I will share and then you will start again, please. Just a minute.
Okay. I think now it's working, Vanya. It's transmitting, I think.
No, okay. The YouTube, yes, but not the.
Okay.
I think now you can start now. I hope it works.
Okay. So I'm sharing it.
I actually cannot see the screen from Adilson anymore.
It's okay because I think he's
it's loading, yeah.
Yeah, okay.
Okay, so now I can share. Actually, Vanya, you need to stop your sharing to me to share my screen.
Okay.
Because, yes.
Oh, yes. Sorry, I think I mixed. No, okay. Sorry.
It's okay. I think now it works. Let me see. Yes. Now you only need to put your screen in full screen, and I think it's okay for the transmission.
Need to put it
Nos 3 pontos, você pode ir ali em tela.
Okay. And now, I think now it's okay.
Yes.
Okay, sorry. Okay.
So, do you have the green lights to introduce the jury members?
Yes, for me it's okay.
Broadcasting is okay. Okay. So, good morning, everybody in Brazil, and good afternoon, everybody in France. We are very happy to be here for the thesis defense of Adilson Roberto Locali Pereira from University, Sao Paulo State University and Nantes University. This thesis is a joint PhD defense between these two universities in France and in Brazil. So I will introduce very rapidly the jury members. I'm myself the president of this jury, Professor Bernard Hoffman in Nantes University. We have the two formal examiners for your thesis, Pedro Esteves Duarte Augusto from professor in Central Supélec University of Paris-Saclay. Then we have Professor Rémi Sorel from l'Institut Agro Dijon, who is a professor in this university. And then we'll have Mrs. Myriam Dupas-Hubinger, professor in University of Campinas. Professor Guilherme Miranda Tavares, who is also a professor in University of Campinas. And your supervisors, we start with Professor Vania Regina Nicoletti, who is professor at Sao Paulo State University. And then Professor Claire Berton-Carabin, who is Directrice de Recherche in INRAE Institute in France. And Véronique Solé-Jameau, who is an engineer in INRAE, also in France. And your co-supervisor, Adeline Bois, who is chargée de recherche in INRAE in France. So, Adilson, now we leave the floor to you. You have about 45 minutes to present your research, your thesis.
Okay, thank you for the presentation, Bernard. So, hello everyone. Thank you for being here today, watching my PhD defense. My project is called Resilient, Sustainable, and Nutritious Pigeon Pea Protein Fractions as High Potential Food Ingredients. The project project was developed in a joint program between São Paulo State University and Nantes University in France. And during my stay in France, I was hosted by Inhae in the ESD team. And my supervisors are Vânia Regina Nicolette, Roberto Carvan, and Adeline Bois. So to start, I will give a brief context about the motivations of this research. So according with data of the FAO, around 95% of the world's food needs are provided for by just 30 species of plants. So this number is very small if you take into account that we have around 30,000 plant species that are edible in the world. In addition to that, if you think about plant-based proteins that have been used to develop new products to the market, we will see that soybean is a global legume crop that has been used, and there is still a lack of food diversity in the products that we can find in the market. So this shows us that we need to further diversify our sources. And when we talk about post-crop seeds, there is a group of foods that are still considered underused in the world, or also called orphan crops. And these species most of them present highly drug-resistant characteristics that's very important in the climate change context that we are facing currently in the world. But the production are almost entirely restricted to small-scale farmers, and they have received very little attention of different constituents of the current agriculture system, like researchers, private companies, farmers, extension servers, policymakers, and decision makers. So Pigeon is part of this group, the foods that are still considered underused. It belongs to the Fabaceae family, and its seed composition presents a protein content that can range from 18 to 28% in dry basis. So it's considered a food very rich in proteins. And this species is native of emerging countries in Africa and Asia. And today, India is both the largest consumer and producer of pigeon pea, accounting for around 90% of global production. And the seeds were also introduced to the Central and South America by the slave route from Africa in the 17th century, and it adapted very well to the different conditions. But in countries like Brazil, the seeds are mainly used for animal feeds. But this is starting to change since some public companies like Embrapa have been showing interest in developing new cultivars that are more suitable for human nutrition. And thinking about the work production from pigeon pea, here we have a graph that compares pigeon pea production with the other foods. According with data of the FAO, pigeon pea is among the most important foods in terms of global production and consumption quantities. But if you take a look, you'll see that the contribution of pigeon pea is around 6% of the total food production across the world. And the number is very small yet. And if you want to change the status of pigeon pea from an underused crop to a more used crop, you need to bring more scientific knowledge about its seeds and potential ingredients, and that way promote some innovations in this segment. So in the first stage of my PhD, I did a literature review about works that was published regarding pigeon pea. And I saw that the protein fractions is the type of ingredients that is most studied. Around 32% of the articles that was published involve some process of protein fractionation. And if you take a look about the protein extraction methods, we can see that most of the works focus in the classical methods that consist in the alkaline extraction of the proteins, followed by the electric precipitation. And other methods also involve some precipitation steps, and this can alter the conformation of proteins. So there is a lack in the literature of processes that are more milder that extract the proteins in the most native states as possible. And regarding functionality, you'll see that around 16% of papers focus in the studies of functionalities of pigeon pea protein fractions. But most of the works focus only in a screening of different functional properties. And I found only 2 papers that focus in a more in-depth study of a specific functionality. It was the case of these 2 papers published by a group from Argentina. One focused in the emulsifying properties and the other one in the gelling properties of the protein fractions. There is no report for foaming properties, for example. So in my work, I used 3 different protein fractions, starting by one fraction that was obtained by the classical precipitation-based method, passing through a method that I use for yield extraction without precipitation step. And the last sample involved the yield extraction followed by purification by chromatography. And regarding the analysis that I conduct, we can divide it in three research axes. So the first one corresponds to the composition. And here, not only the composition of proteins, but also the composition of carbohydrates, lipids, and minor compounds that may be present in the fractions. Other important axis was nutritional quality that was accessed by mainly the impact of some seed pretreatment and processing in reducing some anti-nutritional compounds like trypsin inhibitors, for example. And finally, functionality. And here I first made a screen of different functionalities, and at the end I focused in a specific one that was the foaming properties. So about the objectives of this work, it was to extract, purify, characterize, and evaluate the functional properties of different protein fractions from Pigeon pea. And to do that, the work was divided in three parts. So the first part consisted in a study about the effects of some pretreatment on the composition and functionality of Pigeon pea seed ingredients. The second part was focused in the development of a preparative scale purification process to purify Pigeon pea 7S globulin. And the final part was focused in the air-water interface and foaming properties of crude and purified globulins. So for the first and second part, I used commercial seeds from cultivar. And in the final part, I also used some seeds of per line that was donated by a brother. So the findings of this work, starting by the first part. So this part was already published recently, so I will focus only in the main results that are important for the rest of my presentation. And here the idea was to evaluate soaking and boiling pretreatment of Pigeon pea seeds and their effects in composition and functionality. And when I take a look in the literature about works that focus in pretreatments of Pigeon pea seeds, I noticed that most of it focused only in the production and evaluation of flowers. And there's only one paper that focused in the effects of these pretreatments in the protein fractions. Also, regarding composition of Pigeon pea-based ingredients, I saw the same thing. So we can find more works that focus in the characterization of flowers and only two papers that focus in a more in-depth characterization of the approximate composition using different techniques like chromatography, for example, to characterize these fractions. So there is a lack of studies of this kind from protein fractions for Pigeon pea. And as we saw in previous work of our group, the non-protein compounds are important in the functionality of these ingredients. They need to be considered. And so here we saw two main legs that was to evaluate the effect of seed pretreatment in the functionality of protein concentrates or isolates from Pigeon pea seeds. And also there is a lack of more in-depth studies of the effects on composition. So the main question that we wanted to answer in this part was, how does seed pretreatment affect the composition and functionality of Pigeon pea-based ingredients? So in these images, you can have an idea about the procedure that I conduct. So I had one sample that was the control, consisted of raw seeds without any pretreatment. The soaking was conducted against distilled water for 24 hours, and boiling was conducted for 30 minutes after soaking. So after the pretreatment, the seeds were dried and milled to produce the flour, and the flour were used to produce the protein fractions using the precipitation-based method. And here we can have an idea about the effects in the approximate composition of the flour and also the protein concentrate. So regarding the flour, I started by around 20% of proteins. The carbohydrate accounts for 64%, and the lipid and ash fractions account for 3 to 4%. And if you take a look in the protein concentrate, you can see that the protein content increased by threefold with the fractionation. We were able to reduce considerably the levels of carbohydrate, but there still accounts from 9 to 15%, so there are a considerable amount. And I also characterized this fraction by chromatography, and I observed different classes of carbohydrates And some of them, for example, the raffinose family oligosaccharides, can have an interesting effect in nutritional quality. And starch can affect functionality, for example. And about the lipids, I observed an accumulation of lipids from the protein fraction that was used raw seeds. But with the seed pretreatment, I observed a decrease in the levels of lipids, as we can observe here. And in general, the levels range from 4 to 13%, so it's also relevant contribution. And I also characterized by chromatography this fraction, and I observed the presence of neutral lipids, phospholipids, in tocopherols. So the main results of this part were that the carbohydrate and lipid fractions can alter the functionality of protein concentrates, and together they can account for around 30% of the composition of the protein fractions. Also, the protein concentrates obtained from soaked barley seeds show higher starch levels and absence of phospholipids, and these compounds can have an important effect in the functionality of these ingredients. And mainly, due to the complex composition of protein fractions that we observed, it's difficult to evaluate the contribution of proteins and non-protein compounds to the functional property. So with this, we saw the need to have a simpler system to understand and characterize the intrinsic properties of individual protein groups. So this leads us to the second part of the research that was focused in the development of a protocol to purify Pigeon P7S globulins in a preparative scale. So here, since we know that the composition of the protein structure of Pigeon P presents a highly complex, we can find different protein groups, presence of free lipids, pigments, phenolic compounds, and other compounds. And since we want to study the intrinsic properties of a target protein, in the case, the 7S globulin fraction, so we need to first eliminate or reduce the interfering compounds. And to do that, we can use the liquid chromatography techniques at purification. And that way, you'll be able to separate and purify our target protein. So regarding Pigeon P, some works in the past focused in the separation and purification of 7S globulin using analytical scale chromatography. It starts with a group from India that published 3 papers regarding the purification of Pigeon P globulins. And more recently, other groups also studied about this separation of this protein group. And the main findings that this work brings was that the 7S globulin is a trimer of molecular weight around 108 kilodaltons. These proteins were also identified as a glycoprotein, and they present a high homology with soybean 7S globulin. But we also identified some lacks in this work. So the first gap was that all methods developed used only analytical scale chromatography. Proteins were also extracted using some precipitation steps, and as I discussed previously, it can change the conformation of proteins. And the functionality of purified proteins was not evaluated since the yield is very small from these procedures. So as far as we know, no procedure for preparative scale purification of Pigeon P proteins has been developed, which will be of interest for functional property studies. And the main question that we wanted to answer in this part was how to separate Pigeon P 7S globulin using a preparative scale purification process. So the process that we developed at this part consisted of a 3-step liquid chromatography protocol. This process was first developed in an analytical scale and so optimized and upscaled. And the first stage of this process consisted of the injection of a mildly extracted protein at pH 8 in a desalting column. And this step was important to remove salt and minor compounds like phenolic compounds that are present in the extract. So here the protein is passed through a porous resin, and the small molecules are retained in this resin. And at the end of this stage, we collected a desalted extract. So this desalted extract was injected in an anion exchange column that was packed with a positively charged resin. And that way, we separate the proteins based on the net surface charge. And at the end of this stage, I collect the 7S globulin fraction. So this fraction was further purified by size exclusion chromatography. And here the column was packed with a resin with a wide range of molecular range. And that way we separate the proteins based on size. And this procedure was conducted using two different approaches. So in the first process, I used also dialysis steps between the chromatography steps 2 and 3, and after the step 3 to remove the salt and concentrate further the proteins. And the dialysis was conducted against ammonium carbonate buffer for several cycles. And the other process consisted in the direct injection of one color to the other without dialysis steps. And the removal of salt occurred in the final step during the geofiltration chromatography. So the column was equilibrated with ammonium carbonate buffer, and that way we separate the proteins based on size on the same time that we remove the salt. So the purified fractions in both processes were characterized in terms of proteomic analysis, SDS stage, intrinsic fluorescence, trypsin inhibitor activity, and protein carbohydrate composition. So starting by the first process with dialysis steps. In the first process, in the desalting, I got a chromatogram with 3 different peaks. Being the first one, the peak that is related with the protein-rich fraction, as you can see by the SDS stage result here, that we have more proteins. And in the second and third peak, we did not observe much proteins in the SDS stage, and we attribute these peaks to the minor compounds like the salt and phenolic compounds. So I collect the first peak and I injected it in the N exchange column, and I got a chromatogram with 3 different peaks too. So the first one corresponds to the proteins that were not bound to the chromatograph column, and as you can see in the SDS stage, it is composed of different protein bands. The second fraction, consisted of the 7S fraction from Pigeon, as you can see by the bands in the SDS stage. And the final peak corresponds also with some bands of the 7S fraction, but very less intense. So we collect the second fraction and I injected it in the geofiltration column. And here I got one main peak, as we can see, and other small peaks that correspond to some aggregates. And one interesting thing here is that we observe the partial hydrolysis of the 7S globulin from Pigeon, as we can see by some products that appear around 25 kilodaltons that wasn't there before, as we can compare with the other SDS stage results. So with this first process, I recover a partial hydrolyzed 7S globulin. I will explain a little more about the possible reasons to that, but first I will present the second process. So in the second process, without dialysis steps, I got the same 2 results from the 2 first steps, but with the direct injection of the extract in the geofiltration column, I was able to maintain the protein in their native state, as you can see here, comparing with the other SDS stage. And that way, with this process, I got the native 7S globulin. And the 2 bands that we can observe in the SDS stage, they were identified by proteomic analysis as the alpha and beta chain of Pigeon beta-conjugating. So regarding this proteolysis that we observed in the first process, the reasons for that are unclear to us, but here I put some points that may explain what happened. So the first one is that the 2 bands that we observed around 25 and 20 kilodaltons came from the alpha chain of Pigeon 7S globulin, according with the proteomic analysis of the samples, while the beta chain was resistant to hydrolysis. And we got a similar phenomenon in the literature, when some authors also observed the proteolysis of the alpha chain of soybean 7S globulin, while the beta chain also was resistant to hydrolysis, even in matrices. And according with the authors, this may occur due to the presence of some protease in the flour and protein fractions. So with that, we try to identify accessions that exhibit some enzymatic activity by proteomic analysis in our samples. and identify the aspartic protease in the leucine aminopeptidase that could cause this. But given the alkaline pH conditions that I used, that was around 8, and also the fact that I add EDTA to the process, it's not expected this protease would find optimal conditions of action. So we studied this hypothesis. About the cleavage position, Wilson and co-workers observed the cleavage of P7S globulin around the coping domain, generating products of 19 to 28 kilodaltons. So it's in the same range of the events that we observed from Pigeon P, which means that probably this cleavage can occur in around the same domain. And other important thing is that the hydrolysis that we observed was not observed in the analytical scale of the process, suggesting that aspects linked with the upscale of the process, like a higher concentration or higher process time, may be related to optimal conditions for the action of this protease. So regarding the yield, here we have a figure that shows the distribution of dry matter during the process, that is represented by the different size of arrows, and also the distribution of protein content, that is represented by the different intensity of color. And at the beginning of both process, we started with around 200 grams of initial flour with a protein content of 19%. So that way, at the beginning, we started with around 39 grams of total proteins. But according with the SEHPLC in the naturation condition analysis, we observed that around 86% of total proteins are soluble in the initial flour, and also that 48% of soluble proteins correspond to the 7S globulin. And that way, at the beginning of the process, we had around 16 grams of 7S globulin. They are extractable. And at the end of the process, we got purified fractions with a protein content of around 89% in dry basis. And if we consider that all the mass that we recovered corresponds to the 7S globulin fraction, we can calculate the yield. And the yield range from 23 to 29% of 7S globulin that were recovered. So it can be considered a very good yield if taking account the amounts that are reported in the literature for the recovered of 7S and 11S globulins in lab scale purification process. They range from 1 to 19%. But we only found data from lab scale purification process, no for preparative scale ones. So regarding the composition, as I said, the protein content of the fractions was around 89%. And we also identified from 2 to 3% of carbohydrates in both fractions. We didn't quantify the lipid because it demands a lot of samples to do that, but we expect they are present in low levels in these fractions. Regarding the protein composition by SDS stage, we can observe here the characteristic bands of the native and partial hydrolyzed 7S globulin that we observed previously. But the most important thing is that if you take a look in the low molecular weight proteins, we can see that the process with dialysis step, that is this one, seems to be more efficient in remove these other groups of proteins in the samples than the process that we don't use dialysis. Regarding the carbohydrate composition, we identify a high mannose content in both fractions. And this result is very interesting considering that even the fraction that was subjected to extensive dialysis cycles, we got the same result, which means that probably this mannose is strongly bound to the proteins and can be linked with a possible glycosylation of the fraction, since Pigeon P globulins are known as glycoproteins and mannose can play an important role in it. I also probed the trypsin inhibitor activity of the fractions. And again, the process with dialysis steps was more efficient in decrease the trypsin inhibitor activity compared with the process without dialysis. But we can consider that both levels are low, what is good for the digestibility of these fractions. And finally, I also probed the 3D emission excitation for size matrices of the flowers. In this analysis, I used the powdery sample, so in the dry state. And this This method measured all the compounds present in our samples that present some fluorescence. In the case of Pigeon pea flower, for example, we can see two different regions. One that corresponds to the fluorescence of proteins, and probably they are linked with trypsin, tryptophan, and tyrosine. And the other region are related with the emission of unknown compounds that, according with the literature, can be a mix of phenolic compounds. And the most important thing is that with the desalting step of our purification process, we were able to remove the second region, as you can see in the purified fraction. So it's a good way to show the purity of our samples regarding these compounds. And the other important thing is that it seems that the fraction that was partially hydrolyzed presented a shift in the most intensity peak, which can mean that the tryptophan exposure change with this process. So I also probed the solubility of the sample, since they are very important from the functional properties. And I probed it in a different pH and at low and high salt concentration. So at low salt concentration, we can see a clear effect of pH on the solubility, where proteins presented the classical U shape. And here we have a higher solubility values in the extremes alkaline and base and acid, and we observed lower values around the isoelectric point of the proteins. When we increase the salt concentration, we observe that the native 7S globulin presented solubility higher than 90% in almost all pH that were evaluated. And other important thing, the first one corresponds to the equilibrium stage. And here, all the surface active compounds that are present in the sample start to migrate to the interface to form the interfacial film. And at this point, we can study the absorption behavior of the samples by plotting the surface tension over time. So here we have the results of the at low protein concentration and high protein concentration. And the main conclusion that we can get from this is that we observed a faster absorption of crude protein extracts. We can see it in the green and orange lines here and here. And this can be due to the presence of proteins of low molecular weight and also these non-proteinaceous compounds like phospholipids, for example, that tend to migrate to the interface more quickly than the large proteins. And in a lot of different moments of my research project. And also to the study agents, so to CAPS in Brazil for my PhD grant during 3 years. And also to France Excellence CFR program at Campus France for my PhD grant during my stay in France. And to my supervisors, to Vania, Claire, and Adeline for helping me in everything, basically, for being very patient with my learning and for all the knowledge that was shared. And also to Veronique and Joël, because they help a lot with the purification part. That was very important, a very important part of the work. And to you all that are watching, thank you very much.
Thank you very much, Adilson, for this very clear presentation and within the time that was allocated, that is about 43 minutes. Now, if everybody is okay, we will start our question time session, and we will start with your first formal examiner, who is Professor Rémi Sorel from Institut Agro Dijon. Rémi, you have the floor now to ask your questions and initiate the discussion with Adilson. Thank you.
You want me? Okay. Okay. Thank you, Mr. President. So first, I would like to thank Claire and the other supervisor for their invitation to evaluate this work and the invitation to participate to this oral defense. Thanks a lot also, Adilson, for your nice and very clear presentation. I was very impressed by the quality of your illustration. So your work is, I think, a significant and complete contribution to the knowledge of specific fraction, protein fraction of a promising legume crops. And in your work, you provided an impressive analytical work. I think that this is very impressive. And what I appreciate is the detailed analysis that of the composition of the different protein fractions you obtained, which is scarcely done in in plant proteins today. So overall, I learned a lot, and I was very interested by the proteomic approach. And regarding your manuscript, it was very well organized, and the general discussion was very interesting and useful. Now I have a lot of different questions regarding the composition of the grain, the composition of the protein fractions, and you obtain it. First, my first question is the question about the size of the grain, because you have I read somewhere that the size is 2 millimeters thick. It's right? Mm-hmm. It's very small grain compared to other grain we have. And when you use the grain in your work, you don't so you don't use de-yielding, so you use the entire grain. Mm-hmm. So I would like to know the proportion of the whole in the floor. So an idea of the proportion of this compound.
Of the, you mean in the coat of the seeds, or in the? I didn't got the final part. It was related with the husk, or not?
I was surprised that when you extract, and when we look at the protein concentrate you obtain by alkaline extraction, isoelectric precipitation, it was a very low content in protein, because in general this process is used to produce commercial or industrial protein isolate. And in your case, the protein content was only around 64%. Mm-hmm. So for me, it was very low. And how do you explain that? Compared to other source of other legume source, for example, it looks very, very low.
Yes. Well, I was also expecting a higher value when I saw this, but I think that it can vary with the different seeds and also maybe different cultivars, and also within any protein factor that we use. So for Pigeon pea, for example, a lot of works use 6.25. So this leads us to a higher protein content that is usually reported in the works. In my case, we try to use the 5.7 that is also recommended to plant-based ingredients. And we think that's the way to not overestimate the value of these ingredients. But still, I observed that higher ash content as well, because I didn't use any dialysis steps from this fraction. So I think can be related with the fact that we did not remove as much as the salt as we could, and it could also help. And the fact that I didn't remove the lipids of the fractions at the beginning. Usually some works also the fat, the flour. And in the case of Pigeon pea, we consider that since it presents a low value of lipids, it was not necessary, but in fact we observe a higher concentration or accumulation of lipids in these fractions. So it can also be because of that. So I think it's part because of the methods that have been used that allow us to have this value compared with the ones that we found in the literature, and maybe some variations regarding the species in specific and other cultivars, because we try to quantify everything, and in some cases when we don't quantify the carbohydrate fraction, for example, and we calculate by difference, this can also, let's say, give an idea that's less accurate about the composition that we have. So I think that it's a combination of factors of the methods that are used that allow us to have a better idea of the composition, but maybe some difference regarding the species.
Okay, because the protein recovery was only 20% in this process, so it's very low. It's also very low. So, what do you mean when you precipitate the protein? Because you have a lot of non-protein compounds, so it means that they are associated to protein or to be recovered in the final concentrate. So, because if you precipitate protein, so it means that you have other compounds that precipitate too.
Yes, I think that maybe in Pigeon pea we can also find the significance of other protein groups that were not precipitating at these conditions. Since I use the mainly focus in the globulins, but maybe they also can present significant levels of albumin that will be present in the side stream of the process, and other protein groups, or even aggregates that are present that make it the protein less precipitate at this condition. And also to the fact that we observe that not all the proteins are soluble, that this is expected as well. And in the case of Pigeon pea, we had around 13% of proteins that are not soluble, extractable. So I think it can be regarding the different groups that are present and the way that they will be extracted or not, these conditions that are used.
Okay. In your protein fraction, you never detect the presence of 11S glycinin, or because you mentioned there is 11S, so it's a literature study, and glutenin also, but we never seen the presence of this kind of protein. So what do you mean? They are retained in the
yes, when I saw in the
insoluble part, or
yeah, I think it can be present in the insoluble part. In the proteomic analysis, we also identified some accessions to 11S globulin, but I think they are maybe present in very low levels, and it cannot be seen much in the SDS stage results that we obtain. But in the literature, a lot of work say that the total globulin content from Pigeon pea is around 60%. And we saw, at least with my results, that we observed that about half, 50% correspond to 7S fraction. So I believe that maybe we have 10% or something like that from the 11S. But I'm not sure, maybe they are not all entirely extractable, or in this condition it was not possible to separate them. I mean, regarding the purification.
Okay, I have a question regarding the presence of lipid, because you evidences an enrichment in lipid compounds in the protein fraction, protein concentrates. So, have you an idea of the structure of the lipid that are present. All lipids are organized. It means that you have micelle or you have something else. I don't know, nanoemulsion. I don't know.
Yeah, I didn't identify this in this work, but I think it's important from the functionality, because depending of the structure that can help, for example, in foam stability and other properties. So it's important to have an idea of this, but I don't have much idea about the lipids in my samples. But it's a nice
okay.
It's a thing that must be investigated, I think.
In the case of the purified native 7S or hydrolyzed one, do you exclude the presence of lipid, or they could be present?
I think that possible at the beginning, after the desalting step, but after the amino exchange column step, I don't know, because I think that may be present, but at very low levels. So I don't know if there will be like a significant effect at the end. But well, I quantified the total lipids in the crude extract, and it was around 8. And after the ion exchange chromatograph, I expect that they are removed, but I don't know. And in the also in the proteomic analysis, we identify some accessions related to the action of some enzymes linked with lipids, but I don't know if it can be caused by this or not.
So the limit for you was the quantity of protein material to analyze the lipid content. There is no techniques that allow to analyze small lipid content in or small from a small sample, or it's not possible by HPLC or something.
Yeah, we didn't try it, but it's something that I was thinking about, at least to have a qualitative idea, I think. But for example, from the method that we used, it's necessary 3 grams to do a triplicate. So it's a lot of samples to regarding the purified ones that we obtain from 4 to 5 grams. So we use everything only to do this. So it was not possible, but maybe it's an idea to develop a method, I diluted a little of the fraction, some solvent, and tried to analyze the chromatography, but we didn't try. See.
I have some question regarding the structure of the composition of the purified fractions. So you said that the fraction the fraction was native. So what do you mean when you say they are native? Because what is native for protein?
Yeah, it depends on the structure, right? Yeah. Well, we have an idea about native molecular weight. It's an analysis that I didn't include because we will redo actually for publication. But it seems that the native molecular weight of both fractions are the same, in the same range, around 180 kilodaltons. So we assume that we don't have much change regarding about this. But I call it native only because of the events that we observe about the subunities, that it's characterized from the 7S globulin according with the literature. And we observed some different products that it was not reported before, but it was only based on it. But now, as I said, we intend to improve the discussion and the results with other analyses regarding the structure of proteins. And that way, I think it will be better for publication to show what is native and what is not. So the results will be complemented. But the way that I put now, it was only based in the different subunits in the product.
Because you suggested that the quaternary structure is trimeric, but you never demonstrated that. So
yeah.
You use size exclusion chromatography in denaturing conditions, so you cannot attest that the trimeric structure. So why you don't try to use size exclusion chromatography to see the trimeric, so the multimer?
Yeah, in fact, it's one of the analyses that we will do to complement is the size exclusion chromatography with refractive index detector, basic. But we plan to include this the second month, and also the SAX analysis as well to have an idea about the structure. So there is no angle X-ray scattering. And but at the point that I did the research, it was not possible to determine the molecular weight in native condition by size exclusion because we had a problem with the equipment. But we plan to complement this information as well.
Okay. So what is not completely clear is the molecular weight of the monomer, because sometimes when I look I can so the alpha chain was around I remember, so it was it was about 52 kilodalton somewhere by size exclusion chromatography, and the beta chain was 38. But it's not the case for all the SDS page analysis where you have difference. So sometimes it's not clear. So can you attest about the real molecular weight of these fractions?
We observe a difference in the SE-HPLC analysis with the other ones that I conduct regarding the molecular weight. We don't know yet what it could be. We need to check the molecular weight of the SE-HPLC to see if it's right or not. But in fact, there is a difference in the works of the literature in the lab scale purification process on other groups. They observe bands a little different. For example, the alpha chain was a little higher, around 70 kilodaltons, but the other ones was around the 50 that I observed. I don't know, maybe it can be some related to the aggregation or difference in the cultivars and of the seeds that I used and the seeds from other works. But from what I saw, it's in the range that is usually attributed to 7s fraction. But with some methods, we saw some difference regarding this that we still need to check. Maybe it can be a problem with the molecular weight indicator.
I have one question about proteomics, because I wonder if it was not possible to estimate the percentage of the percentage of the abundance of each protein with this technique.
Yeah.
Because you gave the main protein you can observe, but you don't give the relative percentage of each one.
Yes. Well,
I think it's possible with proteomics to evaluate the.
I think too, but I think it depends on the detector. Maybe I'm not sure if the detector that we use allows this quantification. The only thing that we got was the number of the peptides that were identified and the number of spectra. So for me, we can have an idea about what we have most in our samples, but to really integrate the peaks and quantify, I'm not sure if the method allows this This method was conducted by a group from IHAE, and I only analyzed the final results. But from what I saw, it depends on the detection of the detector, and if we can integrate the peaks and have an idea about the quantification and percentage of each group. So I'm not sure if the method allow go further to have an idea of different proportions. The only thing that we extract was the number of spectra, and from this we can have an idea in what we have moves or less. But,
okay. So now I have a question about solubility and zeta potential, because and the effect of ionic strength when you use 500 millimolar of sodium chloride. So how do you explain the rise of solubility by using salt? Because you use salt after to study the forming property, and so I suppose it was because the solubility rise, but how do you explain that?
Yes, I think that when we increase the salt concentration, the ions help the proteins to bind with water, so we favor the ionic interactions between protein in the medium. So it helps the solubility to increase. And in the case of Pigeon pea proteins, we observed that for some pH, the ionic exchange was more important. The difference between the low and high salt concentration was higher. But in some cases, like pH 8, we didn't observe much difference. I think it depends on the exposure of the residues of the amino acids, basic and acid at each condition, and the way that they will interact with the ions of the salt in this condition. It can help to increase the solubility or not. And in the case of for example, the pH 8, that was the one that I used from probe the forming properties, we didn't observe much difference between the solubility. It ranged from 80 to 90%. And I think that when we have a clear effect of the salt, it's in the range that I used, that was 50 millimolar to 500 millimolar. It's in the range of several foods in the industry. But when we start to go higher, like higher than 500 millimolar, we favor the salting out effect of the salt, and that way the solubility will be lower because the ions will compete with the proteins to interact with water. So in this range that I use it, I think it was suitable for the possible application in food. And in some cases, we observe more differences that are probably related with the way that the structure of the proteins are exposure to the medium at that pH. Thank you.
Okay. Regarding forming property, because forming and interfacial properties, you use pH 8 and 500 millimolar sodium chloride. So, do you think it is realistic for study these properties, because the salt concentration is very high and the pH is not representative of food products? So why do you choose this condition?
I choose this based in a work that they worked with quinoa proteins, and I also use this condition. So I use the same to have at least one to compare, because there are only a few of works that evaluate purified fractions, for example. And since I determined the extinction coefficient at high salt concentration, and the solubility was also high at higher salt concentration, I used this to make it easier to my procedure during in the lab, because it was very at the end of my stay in France, so I need to save some time to do this. But as I said, we should take a look in the low ion exchange at pH 8 and high. We will see that the solubility changes very little. So I assume that each ion exchange between 50 and 500 millimolar, you have a good foamy property. I don't know. I need to prove to make sure, but since the solubility is not changing much, I think that the foam stability in this range will be also good. So I use this because, as I said, determine the main properties of this condition. So it was easier to plan the experiment. But I believe that they're also reproductive in the similar conditions or even with lower salt concentration.
I don't know if I have more time for a question. I don't know.
Yeah,
President.
You have about 5 minutes max. Yes.
5 minutes? Okay, because I have a question regarding the extraction process. I wonder, so could you explain why anion exchange chromatography is necessary in your whole process? If you don't have, if you keep only, I think size exclusion chromatography maybe could be enough. I don't know.
Yeah, I think that the ion exchange is good to better separate the proteins, especially because we usually found some aggregates in it. So if I use only the size exclusion, I think that the separation will not be so good because probably I will have a lot of mixed proteins in the fractions. And when you use techniques like the anion exchange, it's more suitable for globulins. There are proteins that present a lower ionic isoelectric point. And also regarding the yield, it's better to start with ion exchange to recover more powder, and after some works call it polish. So you inject again in the jar filtration to remove aggregates and other things to improve your purity. But in our case, for example, it was good to optimize the process first in ion exchange because we can change more parameters. So we can change the buffer condition, the pH, the increase of the gradient, and in the size exclusion, we can change, the size of the resins cannot change, only the buffer that we injected in the pH. So I think that the anion exchange is better to optimize the process because you can change more parameters and you can separate more the proteins, and after you use the size exclusion so to polish the fraction and eliminate aggregates. But I think that using only the size exclusion, in my case, that I observe a lot of aggregates in the sample, I think it will not be able to separate much the proteins.
Okay, I have a last question. Do you think realistic to extrapolate the chromatography technique at a pilot or industrial scale? And have you some example of plant proteins that are produced by chromatography?
Yeah, for what I saw, only for pea, and I believe that even pea, a few companies may be focusing this process to separate like the 7S and 11S. But today, I think it's not real. The yield is very low and the process is expensive. So I think it's not very attractive to companies to do all this, to separate the fractions. But thinking about the market and what I saw in the papers, I think it's only a few companies that focus in the only example that I saw was for pea, but I'm not sure if it's like a lot of companies that do that or not. It's soybean, actually. I'm not sure if it's for pea or soybean, sorry. And, but for pulses in general, I think that there are still, today, I think we don't have enough knowledge to improve yield and to make the process be suitable for kind of applications, like a final ingredient, for example. I think it can be used the way I did, only to make studies about it and to have conclusions about the seeds that you have.
Okay, thank you, Adilson, for your answer, and congratulations again. So, Mr. President, finished.
Thank you very much, Professor Sorel. Now, if you're okay, Adilson, we'll move with the next reviewer, and it's Professor Pedro Esteves Duarte Augusto from Centrale Supélec, Université Paris-Saclay. Thank you.
Thank you. So first of all, thank you a lot for the invitation, because it's very nice to have this opportunity to meet the friends and discuss about a nice project like this one, and especially a thesis which was made connecting Brazil and France. So for me, this is also special. So thank you for this. As a general view or general things to say about the thesis, I think the topic is interesting. The approach by how you choose to develop it, I agree with it. It was very nice. I like the text. I think it was very well organized and easily to be reading. And what I like specifically, I wrote, I think it's the structure you choose to present the chapters. So to present the kind of hypothesis, so the questions, the main results. So in one page, you can have a nice idea about why that chapter is there and what is the next steps based on this. So I think this is very nice. And I'd like to make some questions and some considerations, some more focus on process engineering, which is my background, so a different view in terms of the chemical and biochemical view of the other colleagues, and also some general aspects, okay? So for example, and I know, so I have some questions, but I know all the time during a project, we need to make some choice. When we do some choice, there are positive and negative aspects. What we need to assure is that the positive aspects are better or bigger than the negative aspect. So based on it, I had some questions to try to compare. So starting by the title, I like the title, but I think it's quite ambitious. So when I saw it, I expect a little bit more than learn about proteins. And in fact, this starts to be my first question. So how you see this project in relation to the ambitions you present from the title and then the introduction and etc. If you fulfill all the idea you had, or if it's part of what you could do and you had more things planned before that you could develop. So try to tell me a little bit about this historic aspect or the chronological aspect. Because, for example, if I look chapter 3 and chapter 4 and 5, they are in the same thesis, no problem with them, but I see two different views of it. Maybe it's Brazil and France, maybe it's something more that you were not able to develop. So first of all, try to make this context for me, please.
Yes, thank you for your question, Pedro. And well, about the title, actually it was a suggestion of Claire. It was not my title, but it was to submit a project to me to go to France, and we need to apply to a scholarship, and we used this title. And I think that at that point it was important because it was a project that was evaluated by a jury of different areas, not only food science fields, for example. So I think it was important to the jury have an idea, because when you say about purification or other things, people don't know what it is. And so, yeah, it was audacious, I think. But at the end, I really liked the title, and I wanted to keep it until the end. And I think that it's a way to show to a broader audience about the importance of the research. And when they put a title that's too much specific, I think that it's important from the academic area, but from the other people that needs to be impacted by these results, sometimes that will not be called to read it and to know about it. So there's the point. And well, from the title, I think that I'm working with a resilient species, and we evaluate a lot of nutrition, the nutritional quality based on the decrease of the nutritional compounds. I think it's kind of related, but no, like we are expected in the titles of the work. And yes, the first part was different from the other ones because I started to elaborate this at Brazil even before the idea to go to France to do the rest. So it can appear some disconnect at the beginning, but I think that there are some points that link it with the rest. So in this chapter, I think it's the chapter that I most characterize the samples in terms of composition. So all the characterization of the carbohydrate fraction, the lipid fraction are in this. And so we start with a very complex sample. And also in this chapter, I screening different functional properties. And at the end, I knew that the composition was very complex, so I needed to simplify the system. So this is the purification part. And after, I focusing only one functional property. So the way that I see thinking in a shape, for example, I think that we start with the basis of a triangle, and after you're high in the top to more specific knowledge. But I think they are linked in somehow. But the idea with the first part, just to complete, was the project at that time was still open. So I had some ideas with Vania to what we can do with the project. So at the beginning, probably if I continue at Brazil only and not include the time in France, I went to other ways, like more focused in the cheese processing and other things that we could do in the facilities of Unesp. But when we went to France and we saw the opportunity to bring more knowledge about the intrinsic properties of proteins, it was an important way to improve the quality of the work, I think. And so we made some adjustments to do this. I don't know if I was clear, but...
Yeah, I understood. I agree. Again, you need to make some choice, and when you have some good results, you need to go on. You can say, "Oh, but I thought to do another thing." If you have a good result, you just need to explore. The point is some connections that I would like to do. For example, your second word in the title is sustainability or sustainable in a general way, which is a very we can discuss a lot about it, but I'm not sure if the proposal you bring to us connect with this. What I mean, for example, in the first point, and especially thinking about a pulse and etc., I would thinking in this source first as a meal, so for people eating, or to substitute animal protein, all the topics like this. But the proposal is to produce a very specific ingredient whose quantities is very, very small. I don't know, in the end, what is the percentage that you sell, let's say like this, in relation to what you harvest in the crop. So by doing this, you also produce a lot of byproduct, for sure, because you are purifying, and as much you purify this, the other, what we can do with the other fractions. So what's your opinion about this connection and what this partial currents or not? Because another point, you say it's produced, I don't know if in Brazil or in the world, mainly by small farmers or small producers, something like this. So you need to improve it, etc. So this is one demand or one topic you discuss. But again, then you go to a very, very small quantities in terms of application, in terms of the ingredient that you are proposing. I'm not saying it's wrong. I'm not saying this is not a good ingredient, My question is this connection among the different topics you intend to discuss.
Yes. Okay. Yeah. It's a very nice question, indeed. But I think that, as you said, during the process of protein concentration or even purification. We have a lot of subproducts, like the pellet, that is rich in carbohydrate and starch mainly. So I didn't focus on this in my project, but I believe that we can have a way to use these pellets, for example, to extract starch that's present in more levels in the seeds, and not only for pigeon pea, but for other seeds as well. And starch can be other potential ingredients from the industry. And regarding the process, I used some things that can be considered more suitable in terms of processing, that were the fact of a milder process, that we have low levels of side steam, for example. We don't have a lot of water consumed in some techniques. And the process of purification itself, because usually purification is conducted, dialysis is very important to remove the salt, and it demands a lot of water to do that, and energy, and salt, and etc. So one of the process consisted of a lower time of processing, and that consumed less water, less energy, less reagents. So there are some points of the research that I think reach this point about this sustainability. And well, as I said in other question, I think that this process that I use to purify the fractions, there's no aim to have like this fraction as a final ingredient that we can consume in the market, but can help us to understand other fractions that are less purified, and that way I think it's more suitable with the fractions that we can find in the market, in the companies. But it's something that I think we still need to try to understand and to improve. I think that I passed through some points that are important for sustainability, to reduce the co-products and etc. But the way that I see today, there is a lot of process in general to fractionate proteins that need to be rethinking about these core products. But I got the idea. I agree that we need to always think about that, about the impact that we cause to the environmental, etc. But the way that I saw, I think the project was not so focused on this part. So I understand what you said about the title. Don't embrace this part of in the project. But I think in my case, it was only some subjects linked to that regarding processing.
You know, it's not criticism necessarily to don't develop this, but for sure to be able to discuss this is important. Yeah. So I think nowadays, if you propose a new process or a new crop to be exploited or something like this, we need to think not only one product. We cannot convince someone today to go to the field and start to cultivate pigeon pea, because I don't know how 1 ppm of it, I don't know the quantity, will be a very nice ingredient to stabilize foam and etc. So you need to think, look, I had all this palette of things. These will be, you can use to produce starch. These will be fuel, I don't know. So a very complete evaluation. And for sure, I'm here to ask you this and verify what you think about, but for sure you will not be able to develop all those views in the thesis. It's normal, yeah. But anyway, let me keep trying to focus on this, because then to connecting also with the last question that Remy did with this one, do you think if now you have a product that you can add this ingredient and etc., you tell it's very good properties and so on, but the cost benefit of starting this and all this very specific purification process that it's needed? Do you think it's worth, or it's very expensive, or it's a very, I don't know, complex technique for an ingredient that you can just take egg and use, or what do you think about it?
Yeah, I think that this procedure can be, that we developed, the purification and preparative scale, can be important, for example, from some companies. They are interested to better characterize seeds and differentiate cultivars, for example. So it can be a nice way, I think, to characterize seeds from different cultivars and know exactly the amount of proteins that you have from each group, and maybe probe some functional properties to have an idea what is that seed is good to, what kind of applications, what kind of products. And it's a nice way, for example, to other species to have an idea about the proportion of 7S and 11S globulins, and based on it, try to select the most suitable cultivars to cultivation in large scale, for example. But the way that I see for Pigeon specifically, I think that, well, I make some contribution to the food science area, but there is also others that need more attention, like develop new cultivars first. So cultivars, they are more suitable to large scale processing. These kind of things are important to increase the production of Pigeon pea. And cultivars are more resistant to different environmentals, not only to the ones that have been used, like in India, for example. And I think
that question I would like to do. I'm sorry to interrupt you.
Yes.
But yeah, because I developed a work with people from Embrapa, Rodolfo, etc., exactly to select some cultivars. So they started with 200, I think, and then we finalized with 2. So those 2 are the good ones for food production because of this, and that. And then you cited that you worked with them, but in the material and method section, you always say that you just bought the grains into the market. So have you used a specific line or specific variety or etc. Selected by them, or you just bought in the market one grain that you had no background about it and use it? Did you get my question?
Yeah, I think I get it. I used 2 different seeds. So one was what they call it commercial, that is the one that we bought from a supplier in São Paulo. And the other one was the seeds from Embrapa. And from these seeds, we know cultivar, and it was a species that made part of this study that you mentioned. It was one of it. And the other one, I didn't know much about the process because it's cultivar. We don't know more details. Well, we were strategic to use each type of seeds because from the commercial one we had a higher amount. So I use it for the purification process, for example, since it demands more samples to get the reference. And in the final part that we consider that was an important part of the work, that was the proof of the functionality. I used the seeds from Embrapa to compare with the commercial one.
Okay. I'll try to go to some specific questions because time is passing. Especially chapter 3, where we can see the process by itself. So about it, I had basically 3 questions that are connected. So you selected a process, but without changing the process conditions, you know? Mm-hmm. For example, one you make the hydration in one temperature, which okay, the choice, but why this period of time, and then all the steps like this. So my first question is, if you vary these process conditions, would you expect to obtain different fractions after, or more or less the same thing? In this case, why you boiled the grain? In this case, why you dried the grain? Because you could, after hydration, just mill and make the separation as we do, for example, for starch extraction in the industry or something like this. Maybe it's because to have the opportunity to do this in the lab, to send to France, etc. If this is the question, okay. But looking from a process point of view, to then say this is the way to produce this very special ingredient that I'm claiming with the thesis. What do you think about the process conditions and choice you did?
Okay. Yes, about the process, the pretreatment seeds of the seeds and also the protein fractionation. In this part, I based it in a work that was published at the beginning of my PhD that was focused in some seed pretreatment after, before the fractionation of proteins. It was the only work that evaluated further the protein fractions. So we went to use this similar procedure to compare with the results that this work brought. And since we observed that they get a nice yield, and the yield was one concern for us at the beginning, we thought we use the same procedure to save some time and focus in the other steps. And about the boiling, I tried to reproduce one condition that is usually applied to have soft seeds, like to consume, like as I mean. But after my research, I was thinking about that, and I think that in this case, the boiling can be conducted at lower time. For example, not 30 minutes, but 10 minutes or even lower, just to have some effect of the temperature, but not saturate the proteins, for example. So I think it can be a nice idea for future works to change the parameters, or even soaking. That soaking can also be conducted, I don't know, in 40 degrees, 50 degrees, and can have some advantage in terms of remove anti-nutritional compounds and prevent the denaturation of proteins. So I think that I started with a very conventional times that I saw in some works that evaluate this type of pretreatment. But from this, I think we can think about using some variations in the process to see the advantage that we observe in my results, like the reduction of anti-nutritional compounds, and not denaturate the proteins that can affect functionality. And regarding the drying, I dried because I believe that in the flour we have a higher surface area, so maybe it would be better from the extraction of the proteins, but not sure. I think that some works can also evaluate directly from the seed and do the extraction and so on. Can save some time and can be an idea too. But at Unesp, there is other student that I think tried to do this without drying, and she observed that the levels of proteins were lower when you extract directly from the seed. So I think it's something related with the way the contact of the solvent medium with the proteins that is better using the flour.
Because if you increase temperature, if you dry, so talking about sustainability again, there are some unit operations that use a lot of energy. So if you can, and of course, if you can remove any unit operation, it's nicely from an industrial point of view. And maybe you focus your project in a procedure which is basically done to cook and to eat directly this grain. But as you purify a lot after, I don't know if you need to do this in this stage, So again, it's not wrong, it's a choice. But maybe by changing the process conditions and the unit operations that you choose, maybe you can have completely different results at the end. I'll do the last question only because we have only some minutes. It's not clear for me if you choose the foaming properties based in some results and then you say, oh, this is nice, so I'll go through this side, or the opposite.
Yeah, it was a mix of different factors, I think. We have the scientific part, so we see in a lot of papers being published regarding the foaming properties of globulins. They are very useful because they are in abundance in food seeds. But I also had the practical part because I probed the interfacial properties, for example, at the very end of my stay in France. So I need to save some time. And if I use, for example, to study the emulsifying properties, I will need to probe the oil-water interfacial properties. And for that, I need to purify the oil, so I add more steps to the research. So we save some time in it. It was also a personal choice because in my master's I worked with emulsification, and now I want to probe other functional properties. And it was a coincidence that I found some specific works in the literature that focus in the emulsifying and gelling properties, but now in the foaming. So I think it can also have an idea about the potential of PGMP proteins in a functional property that was never probed so in deep, I would say.
So it doesn't close the history. Maybe there are some other hidden, interesting properties for PGMP that can be exploited, correct?
Yes. I think ideally I should probe it for at least the three main ones, that is the emulsifying, the foaming, and the gel. But due my time, I need to focus on only one, so we choose this one.
So to keep the schedule, I finalize my intervention. Thank you very much for the opportunity again. And I pass the floor to the president again. Thank you.
Thank you.
Thank you very much, Professor Augusto. The questions are very rich and very interesting. I will now request Professor Miriam Hubinger from University of Campinas to come with her comments and questions to you, Adilson. Thank you very much.
Thank you, Mr. President. Thank you, Adilson and Vania and the other co-directors for inviting me for taking part in this thesis. It was a very interesting experience for me because I was like Pedro, I was expecting something more on process engineering, and it was in a quite different subject matter. But it's very interesting indeed. I think you have a very good work, a very impressive work. You have a lot of analytical work, and the title, contrary to Pedro, I like the title. I think it's ambitious. He is right when he says that it does not link with some parts of the work, but for me it was quite interesting why Pigeon P is resilient and things like that. And I think you have a very good work, and you had the opportunity to develop a lot of skills that here in Brazil I think you would not. So I'm going to make some questions. First about your work. First of all, I have some suggestions, but I don't know if you can correct this final file. Is it possible to very little things that I think it would be interesting to? I'm not criticizing, but in chapter 1, for example, you have the tables. They are numbered like in the article, but they should have the numbers of the chapter here. It would be interesting if you could change that. The pages are 34, 37, 38, 41. And there are others too. And I have one question in the chapter. I know it's already published, but when you wrote the chapter, you were not aware that you are going to purify the protein and work with the 7S globulin. Is why you don't mention this in the first part of the work?
Yes, when I started to write this review, it was in the very beginning of my PhD. So the idea was focus more in the some seed pretreatment, and protein fractionation methods in general. But not focused in the purification. But at the end, since I was writing during my PhD, at the very end, we tried to include some things about purification of GMP proteins.
Yes, I saw that. I think it's on chapter 4. I don't remember very well, but I saw it. Okay.
But only some small comments about what was conducted with purification of the GMP proteins. But the focus of the review was like a more broader, I think.
Okay. No, the review is quite good, but when you read it, you are not expecting to the part of purification, chromatography, separation at all.
Yeah. Yeah, it's something that came after.
Okay. And I have some specific questions on chapter 4.
Mm-hmm.
The first one, as you work with ultrafiltration, desalting versus ultrafiltration, if I well understood, I would like to know, it's a supplementary figure. I think I'm a little lost here. I think it's figure 4.2 or 4.4. 4.2. How can you see? Maybe I'm not familiar with the technique. How can you see what you have on retentate and filtrate by the emission excitation fluorescence? Because you affirm, you say there that we have some kind of protein there or not, and I would like to know how did you make this analysis?
Sorry, Mira, it was the figure of the supplementary part, or? Yeah,
it's page. Let me
just make sure.
Page 165, a supplementary figure 4.2.
Okay. 4.2. Is it in the DLS or in the fluorescence?
Sorry?
Is the results of the DLS or no? It's the fluorescence, right?
Yeah.
Okay. So here the idea was to try to separate the two areas that we observed by membrane ultrafiltration, by centrifugation. So the first image that we are seeing is the whole protein extract. So in this whole protein extract, we can see the two regions, the one from proteins, that is this one from the bottom, actually the two ones that we have, and they represent the emission mainly of tyrosine and trypsin. And the second one is the region that I mentioned that can be a mixture of different phenolic compounds.
Okay.
And with this tube, we put the extract in the top part, and we do the centrifugation. And that way, we separate in the retentant of the filter and in the filtrate each different fractions of our initial extract. So I analyzed by this measurement each one of the separated fractions, and we can see that the one in the filtrate, that is the letter B, we can see the region of proteins because they pass through the filter, and the other ones, there are the compounds that are retained in the filter of the centrifugal tube, are the ones that we saw the phenolic compounds or other compounds.
Okay.
So we tested at lab scale, we tested two approaches to remove these phenolic compounds. One was this membrane ultrafiltration, and the other one was the desalting. And we got the same thing, but we observed that with the desalting it was more efficient to remove some aggregates, so we used this method. But the idea here was to show that with this, we can also separate the phenolic compounds of the proteins.
Thank you. And this axis that is appearing here is the intensity, the axis that you see outside the figure. Is the intensity that we see here?
Okay. You mean in the
I mean, there's all these axes that are, I don't know, I think it's the intensity of what you are, yeah?
fluorescence intensity, yes.
Okay. And another question that I have in this chapter that I think it was quite interesting is the proteomics analysis. And I think, I don't know, maybe Guilherme will tell us later on, but I don't think, I haven't seen this before, not here in Brazil, not in the thesis that are participate or the thesis that our students are doing. Do you have a library with all the proteins, and then you identify, after separating yours, which one corresponds to the libraries? Is the way that it works?
Yes, and we use we can use the library of different species. In the case, we found one from Cajan, so I use a specific one from the Pigeon. But it's a library that is compare and identify its spectrum.
Okay, and then you decide, you see which proteins are present in your fractions.
Yes, based in the percentage of the coverage and the number of spectra and number of peptides.
Okay, and now linking part of chapter 4 and chapter 5, where you measure the air-water interfacial properties. I can see that the 7s fraction is the best one, I think. You say for producing for formability and even for probability, I suppose, when you put in different ionic strengths. So can I conclude, if I'm going to produce a protein or an ingredient for food industry, that the 7s fraction is the most indicated one from Pigeon P to be produced? Can I conclude that, or it depends on what it's going to be used?
I think it depends on what you're using, and also the composition of the other compounds, because some cases you can have a fraction that is richer in the 7s fraction, but you can also have proteins from other groups. And the results suggest that we can have a lower formability due to the interference of these other compounds. So I think it depends on the level of the percentage of 7s globulin fraction that you have in your fraction, and also the other compounds like lipids and minor compounds. I don't know if we can conclude always that when we have the 7s in this proportion, we will have good properties, because it depends on the composition of the non-protein compounds as well.
Okay, if you are going to establish a process to produce Pigeon P fractions, I think everybody, the two that preceded me, also have made some question about it. Do you think the process can be already established, or you need a lot of studies or other studies to make some conclusion about it?
About the stabilization of the foams, you mean, or in general?
No, I'm not talking only about foaming. I'm talking about, if you are going to produce some protein part from Pigeon P, some protein from Pigeon P. do you think you can already establish the steps of the process, or do you think it's not completely elucidated if you have different, because the yield you have is not very good, no?
Yeah, the yield is not so high. Yes, I think there is still space to more investigation regarding the fractionation method, and mainly because today we see a tendency of the use of dry methods to fractionate protein, and that way we have a better yield and then we better preserve the native state of proteins.
Mm-hmm.
So I think that the results that I got are good to use as a stabilizer in the food system, but there are still space to further investigation regarding different process methods that you can use, like the dry one that I said, or less expensive process in general. And this can affect the conformation of proteins, mainly comparing the aqueous with the dry fractionation. For example, in the aqueous we have the presence of solvent, and this can disrupt the protein bodies and affect the conformation. And in the other way, we don't see this. So I think that not only for Pigeon, but from pulses in general, there's still space to investigate mainly the way that we obtain the fractions.
Okay.
Because some works focus, for example, in modifying the structure of protein, but there's also an approach that says that maybe we can think how to obtain the proteins first, and to don't need to modify them after to have better properties, for example. So I don't know if I was clear.
Okay. Well, I think that these are my questions. I wish you a success. I think you have a very interesting and impressive work. And I finished for now.
Thanks, Emilia.
Thank you very much, Professor Hubinger. I will now ask Professor Guilherme Miranda Tavares from University of Campinas to please address his comments and questions to Adilson. Thank you very much.
Thank you, Mr. President. First of all, thank you, Adilson, and the supervisors for this invitation. I learned a lot during the reading of your thesis. I am learning a lot during the discussion that I'm hearing now. I join my colleagues to congratulate you, Adilson, for the quality of the document that we could read. And also, like my colleague said, this is an impressive amount of work, impressive volume of work, different techniques, and quite interesting work. And like Pedro said, for me, it's also special to be here in this PhD Viva that was constructed in between Brazil and France. It's quite nice. I do not have a lot of time, and I have a lot of questions. I will maybe select some one of these questions, and I go direct to the point. But I believe the approach that you applied in this thesis is very, very updated. Now with my students here, we are discussing a lot of that when we talk about protein-rich ingredients, we cannot only see only study the behavior of the proteins, because several of the different behaviors that we see, the technological properties of these ingredients are actually related with minor compounds that might be present in these ingredients. So your thesis bring very interesting elements to corroborate that. Much of your discussion during the different process and the behavior of the different ingredients that you could obtain are related with some minor compounds that might be present, also related with the for sure the structural properties of the proteins, but the minor compounds might have some considerable impact on the performance of the ingredients. So my first question is in chapter 3. I like as Professor Pedro said, we need to do some choices, and we need to ponderate them. And very often there is no true answer for a path that you choose. But why did you choose to extract the protein in chapter 3 using a pH of 12? It's a quite high pH, and the chemistry of proteins under such alkaline conditions is tricky. So why did you choose to extract the protein in such high pH?
Yes, thank you for your question, Gary. And I did it in a method that I saw in the beginning of my PhD, because at that point there was only a few works of Pigeon, and some used it at pH 12 and got a nice yield. And at the beginning of my thesis, since the project was still open, I was discussing with Vanya about possible ways that we can go. And one of it was using the protein fractions from some application, like in a food or food product, or even microencapsulation. And for that, it demands a lot of yield to prove this kind of things. So the yield was very important at the beginning. So since I saw this work and the yield was quite nice, I thought maybe I can focus in try this to recover more protein. But I agreed with what you said. And in fact, with this pH, we can have the oxidation of phenolic compounds, the oxidation with proteins, and can have formation of some aggregates and things that can decrease functionality. I will understand that better after. So if I need to choose now, I think I will try to use a lower pH, like pH 8 or 9 to the maximum. But it was to save some time and to guarantee that we will have some yield good for the project. Because I try in Brazil, I try a lower pH, and the yield was not, it was a little higher, a little lower, sorry. So we try to fix in this one to guarantee that we will have a good recovery of proteins.
Yeah, it's something like that. We also learned during the process. This is what is a PhD for. Still in chapter 3, you mentioned at the end of this chapter some the possible contribution of saponins to explain some of the data you obtained. Why didn't you quantify the saponin among all the other components that you tried to quantify in your product?
Yes, we discussed about this to quantify, but in France, there are some companies that can do this, but with the time that I had, and it was more about the end of my project, we didn't send the samples to do this, because we could develop the method, I think, but since it demands a lot of time to do this, to probe and to change the conditions, it can be time consuming as well. But I know that saponins are very, it is present in Pigeon in high amounts, and for sure it's a compound that should be further investigated in relation to foam as well, because that is the property that I measured. But I think it was because of the time, and since I was able to do this in the lab, I mean, it depends on other companies to do this. So it was because of that.
My question was a completely honest question, because I'm trying to track in some tips because I also have the same questions in the work we are developing here, and like you said, for foaming, regarding foaming properties, saponins are may be quite relevant on the foamability of the ingredients. So it might be important to also take a look on their behavior.
Mm-hmm.
I was quite intrigued by the results reported in chapter 4, in particular the hydrolysis process. One of the main hypotheses that you had, and you mentioned it during the presentation, it was proteolysis, and you mentioned 2 possible enzymes or group of enzymes are part of protease in leucine aminopeptidase. Regarding the conditions, the physicochemical conditions of the medium, maybe their actions are not the real reason that they are related with the protease you observed. My question, and I really don't know the answer for that, it's a very specific hydrolysis, and I don't know, but are these protease specific protease or more broad protease in terms of actions?
I think there are more specific ones. We try to do some assays to reproduce this in different conditions, and observe some small changes by a stage, for example, but never in the same way that we got with the process. So it seems that it's very specific on this.
Okay. I don't really know the action of these enzymes, but it's not that common protease with very, very specific action sites in seeds. Usually they have a broad spectrum of action, but I don't really have more information about that. But I was really intrigued, and still in this matter, when we see the final sect profiles of your purified fractions, we don't really see a lot of difference between them. Actually, for the native one, we have a little bit more populations at small molecular weight. How do you explain that?
Yeah, I'm not sure too about them. I think that the hydrolysis effect preserves the native molecular weight of the proteins, but maybe it changed other things in the structure part. But I think that for that, I need to do more experiments regarding the structure to try to figure it out. But
did you understand why I'm asking you that? Because when you see the main peak of the two fractions, they are exactly in the same retention time. I don't know if maybe the column are not able to segregate the two populations, but you are working on in denaturation conditions, so we do not really have anymore a trimeric structure that we could have before, and still for the native one, the population of a small molecular weight, they are more intense compared with the other one.
In the same region of the chromatogram, yes. Should be. I'm not sure why we saw this, but they appear in the same position. But I'm not sure how to answer that.
Yeah, it's maybe related to the performance of the colon in separating this population. I don't really know, but I was also intrigued when I saw the final sex, the chromatogram for you to purify the 7S protein.
Because I would expect a higher difference between them, but based on it, we saw in the same position.
And still on that, in page 155, the 3D fluorescence spectra, I was also wondering why the unknown components that we see up right, they are more pronounced or they are more intense. The signal related to the unknown compounds are more intense for sample that was subject to dialysis in the dry state fluorescence.
Okay.
We see the shift on the protein part or the typical protein part, but in the liquid state there are no different in this part. But regarding the dry state fluorescence, the intensity of the signal of the unknown compounds are more intense for the sample subject to dialysis.
Yeah, I think maybe it's related with the processing. The removal of these compounds is more efficient in one of the process. In the other one, we still can see some residues or something.
Yeah, but I don't know. There is maybe a dichotomy on that because we are supposed to have maybe more pure, pure crude after dialysis, and when we saw when we saw this data, it was a little bit different from what I was expecting.
Yeah, I think it maybe can be related with the way that the minor compounds were removed.
Yeah, maybe.
Not sure too.
So yeah, it's another doubt also. It's in the table on page 178. How did you calculate the last row, the theoretical composition of 7S globulin?
Into protein factor?
Yeah, yes, it's the table with the amino acid data, amino acid content data. Is the theoretical composition of the 7S globulin, how it was calculated? Because, so more specifically, why did you consider a 1:1 ratio to calculate that? At the bottom of the table, we can see you use a 1:1 ratio to calculate this theoretical composition. I did not understood.
To calculate this theoretical one, first I retrieved the sequences of the alpha and beta chain that are present here, and so I consider the 1:1 proportion. But it was a hypothesis that we used because we don't know for sure that the real proportion. And I found only one work that based in SDS stage that said that the beta chain, the proportion from alpha and beta chain is 1:2. But based in the intensity of the bands that they observe in the SDS stage. And I don't know if it's the most suitable way to determine this. I think there may be other methods to quantify by chromatography maybe. But since it didn't quantify, we tried to use this hypothesis that they are present.
Yeah, because it is supposed to be a trimeric protein. And here we are calculating a quite of dimeric structure.
It's 1:2.
Maybe, yeah. I did not well understand why did you consider 1:1 ratio, and maybe you should explain a little bit in detail why this decision was made.
Yeah. Okay.
So yeah, I need to ask that. It will be my final question, Mr. President. But it is maybe the simplest analysis you did, but in my opinion, one of the most challenging ones. It's the foaming properties with the syringes.
Okay.
How did you manage to get those very small standard deviation using this analysis? It's not easy at all.
Yes, the use of the software helped it, but it was still difficult. From some, I needed to do more than 3 times and 3 applications. But I think it helped it because otherwise, with the markers, for example, the deviation is higher.
Because our information are from the image treatment with image G?
Yes, image G.
Okay.
So basically ratio of the full volume, and it was easier to calculate that way. Otherwise, it was more difficult to have good deviations.
Yeah, it's not easy. I know it's not easy to have a small standard deviation using this kind of approach. And you did, bravo.
The form is not always so easy because sometimes they can have different shapes inside the syringe, so it's difficult to measure the volume. So we thought that with the software it would be easier too.
Yeah, when we read the methods, it's quite a simple description, but it's not easy to perform this analysis. I know that.
Yeah.
So it was it. Thank you again for the invitation, and congratulations for the document and also for the presentation. Like Professor Remy Sorel said, it was a very clear and well illustrated presentation with a very, very good flow. So congrats. Thank you, and Mr. Presenter, the flow go back to you. Thanks.
Thank you very much.
Okay, thank you very much, Professor Tavares. Before leaving the floor to your supervisors, I also, as an examiner to this thesis and biochemist, I have a few comments and remarks and questions also. First of all, I would like to thank you and your supervisors for inviting me in this very, very, very interesting thesis work. And I was, as it was already mentioned before, impressed by the amount of work that you have been doing during this thesis and about the quality of your manuscript. It was very easy to read and pleasant to read also. And I was also impressed by the way you convey the results and discuss about it. So it is hats off for that. And I was also very impressed by your presentation today. It was very fluid, very nicely illustrated. And so that was very, very nice. And you chose to focus on some aspects of your work during your presentation. And that was very clever and very smart from you to just focus on some of the things there. So aside from that, I have a few questions and remarks. Maybe I will start with a few remarks about your manuscript. Maybe you could provide some additional details on the tables where you have your proteomic analysis results shown, that you provide some additional legend in these table captions, because I felt that it was not present in the materials and methods about what you, for example, what you mean by a group, what is the peptides column, what it looks like. So some information was missing for me, but the fact that you made an effort to provide these tables was very, very nice. And I will maybe focus some of my questions on the proteomic analysis also. And in some cases, I was also looking for additional results, additional details in the captions of figures to understand well. For example, if you take one of your SDS page figures, maybe, for example, page 151 of your manuscript, you will see the bands there. And you mention in the main text, you find two main bands for the 7S globulin. Maybe we could identify them by arrows. Would it be pertinent for that, or what do you think about it?
Identified by what? Sorry, the final part I didn't.
By some arrows pointing.
Yes, could be an easier way to see. I think I can include. Mm-hmm.
Yeah, I think that will help very much to make a going back and forth with the text and the manuscript itself to identify these bands there. Okay.
Yeah.
And for my own, I'm not a specialist of food science myself and the chemistry of proteins in plant extracts. I have a question because on page 150, In the second paragraph, you talk about 7S globulin fractions, and you talk about the alpha-beta chain of 7S beta-con-C-glycinin protein. Are they two different proteins?
Yes, there are different proteins that were. You mean the beta-con-glycinin, or?
Yeah, beta-con-glycinin. Yes.
Yes, this is because there is the 7S family from the different proteins, and inside the 7S family we can find different types of proteins, and beta-con-glycinin is one of it.
Okay. So that is clear. I think that could be mentioned somewhere previously in the text, that in the 7S, what is 7S to start with, and what could be there should be a mixture in the fraction of the 7S, and among these there are the con-glycinin proteins and the globulin proteins. That would help very much, because you mentioned the con-glycinin alpha and beta chains many parts in subsequently. It will help to clarify and to avoid any confusion from the reader who is going to read your manuscript, I think. Yeah. Okay.
Maybe explain that we have the globulins, and inside globulins we have the 7S fraction, and inside this we have the con-glycinin.
Yes. Now regarding the proteomics approach, I did not find in the manuscript on whether you perform the proteomics approach directly on the extracts coming from the flower, that is the flower itself, or is it after? I think in one of the cases you said after fractionation by a liquid chromatography. And is it right, or is there some additional thing that you perform before going for LC-MS?
I analyze it in both ways, using separation in SDS stage. So we cut the bands of the purified fraction.
That was not mentioned in the manuscript.
Okay. So maybe I can include this one. It's because it was two methods. It was one from the gem and the other one from founders. So on the flour and the purified fractions, I evaluated the founder state. And for the purified fractions, I also evaluate the bands on the SDS page. So
that's interesting. It was not mentioned, because I had, this is one of my questions, because if you cut the band there, you might find more easily some precise proteins, and that was my question. In particular, when you look at the table 4.2 on page 152, was this identified after band excision, or is it from another fraction?
In what table? Sorry, in
page 152, table 4.2.
52. So this one is from the whole extract.
Okay, so I think it would be worth to mention in the captions here of the table what is the original, what was the original sample that was used there for this proteomic data that you are showing here.
I mixed it up. Actually, in this table, we have the results of the SDS page. And in the supplementary, we have the ones from the total.
Oh, okay. So exciting results. Yeah, because it has to be the SCH page, because everything is from the 7S fraction. Yeah. Okay, so I think it's worth that you add this information here in the caption of the tables and the supplementary tables about the proteomics analysis. So not too long. Yes, sorry. Yes.
No, I was just agreeing.
Okay. And regarding the proteomics analysis, you mentioned, and it will be my second part of my questions, is about post-translational modification of proteins. You mentioned that your 7S globulin might be subjected to N-glycosylation during the processing of the protein. And I had a very simple question. Did you perform deglycosylation with glycosyl hydrolase enzyme of your protein extract before trypsination, before subjecting for enzyme hydrolysis, and before subjecting it to mass spectrometry? Did you remove glycosyl serum?
Mm-hmm. I'm not sure because we did it in the other lab in Fahrenheit, so other people performed the analysis, and I'm not remember now if they applied this step during the preparation of the samples.
Okay, and before that, on the flour extract, or at some point did you deglycosylate? No.
No.
Okay. So that was one of my questions, because sometimes it is worth doing deglycosylation before doing the spectrometry, because the oligosaccharide itself might be of interest, but it's not the purpose of your thesis. But at the end of the day, it might help on the physicochemistry. Even also the analysis in mass spectrometry might be a little bit more easy without these oligosaccharides, maybe.
Mm-hmm. Yes.
Okay. So my last question will be about glycosylation and the properties of your proteins.
Mm-hmm.
You rightly mentioned that glycosylation might have an impact on the formability and so physicochemical properties of your protein fraction. So did you again treated the protein to remove the glycosides to test this hypothesis or not? Whether there is a...
I used the whole powder only.
Okay. So I think that would be very cheap and very fast to do, is to treat your enzyme fraction with a glycosyl hydrolase cocktail that is commercially available, to test the hypothesis whether there is indeed a relation between the formability and whatever properties you're looking at, and presence of glycosylation there. And you mentioned in your presentation today that there should be an impact on that.
Yeah.
So that could be... Yeah.
It can be a way to observe the effect only of the 7S fraction if we remove the glycosylation, the interfering that maybe the glycosylation has in the functionality. I think it could be a good way to compare both.
Yeah. That, I think if your paper is not yet published, and if by any chance somebody has the chance to do this, to check and compare without or after deglycosylation, that will strengthen I think the argument about the potential role of glycosylation there.
Yes.
Yeah. I was not. I understood the structure of your manuscript, and I found that you were talking about bioactive peptides and things like that. By any chance, during your thesis as a side project, did you check for bioactive peptides?
No. I only saw this when I was writing the review, and I saw that some works focused on it from Pigeon, but it was just a few sections of the review paper. But in the planning of my work, I didn't talk about that. But it can be a nice way to valorize the results as well, to see the importance.
And if it's a more general question, as a general to test your culture, what would you do if you have to do something about bioactive peptides? Or what could be the work that you would perform? Yeah.
I think I will apply some treatment to this protein concentrates that I saw. I think if I'm not wrong, some hydrolysis to get the peptides, and to see the benefits, like in the ingestion of the fractions, if they have some advantage to the absorption of some compounds, something like that.
Okay, some bioassays to check whether they are active or not on some
Yeah.
Okay.
I think.
Okay. So I'm done with my questions, Adilson. Again, congratulations for this excellent work. And I now leave the floor to your first supervisor is Professor Vania Nicoletti from Sao Paulo State University. Please, for your comments and.
Thank you. Maybe yes. Can you see me because I'm just okay. I cannot see me because I'm streaming the screen.
Yeah.
Okay. I don't have questions. Because I expected that in reality I expect that I will talk afterwards the end of the session, but now I have only to thank you and congratulations Adilson about his work, as all of you have said, impressive work. And I am very grateful and thankful for being part of this. And well, let me ask Adilson, what about your plans for the future if you are in this defense? What do you think about how you see your future in research, and maybe you could make some comparisons about your work in Brazil and France, if you can share some of your view about that. And if I don't have the chance to talk again at the end, I would like to take this opportunity to thank very much for the partnership of my French co-workers, Claire and Adeline, on all the support they give to Adilson and our work. And please, Adilson, feel free to share your visions with us.
Thanks, Sonia. Well, about the next steps, I think that I intend to continue like in a postdoc. I think that the next step will be a postdoc to bring more experiments and to publish more papers to in the future be able to apply to a permanent position like in a public or private company focused on research. I really like the subject that I worked in my PhD about the plant-based proteins. I intend to continue in this field if possible. And while comparing the experience that I had in France and Brazil, I think that it was very enriched the period that I passed in France because most everything that I saw was new for me. And Avonia knows that in my master I worked with emulsification, microcapsulation, by spray drying, and it's a completely different universe. So I learned a lot of proteins, and I think that I got a lot of new knowledge about the characterization of proteins. So now I have a more broader view since experiments to obtain fractions, characterize, and maybe an idea of application. So for me, I think it's a full experience. It was challenging, but it was very enriched as well. I think that's it.
Just to add some comments that I would like to share that the first year of Adilson work in Brazil, we were in the pandemics. It were 2020. We were in the pandemic, so there was a lot of difficulties. And also, I would like to thank the partnership from Embrapa. Unfortunately, due to many practical difficulties, we could not extend our work on analyzing further the samples of the bull retrievers that many mentioned. But we of Adilson was a first step, and working with Pigeon P, I think that Adilson could remember that it was motivated by some knowledge I had on this kind of seeds on my experience, practical experience in Brazil, because in Brazil, many of people that lives outside the cities, in the suburbs, they cultivate their own plants of Pigeon P. It's an ancestral culture that I'm very grateful that the work of Adilson could contribute some way to bring more knowledge and light to this culture. And so this is what I would like to share, and thank you again for you all participating this year.
Thank you, Vanya.
Okay, thank you very much, Professor Nicoletti. I would invite now Professor Claire Berton-Carabin to please share some comments, remarks, and any questions with Adilson.
Thank you very much, Mr. President. I'm not going to ask you questions, Adilson, but I'm very happy still to have the floor for a very short time, because there are a lot of people that I would like to thank. You, of course, Adilson, and I will come back to this, but of course I would also like to thank the committee members who accepted the invitation to be part of the committee, and who thank you very much for your time evaluating the work of Adilson. Well, despite the video conference format, it was really great to see the very lively discussion and on many aspects of Adilson's work, and so that was really very nice. Now, of course, Vanya, I would also like to thank you for this for the collaboration that we've had. Adilson is already the second PhD student who could from your lab who could actually come to spend 1 year in our group here. So I think it's it's been a pleasure for everyone here also in the group to welcome Adilson. And then, also with the Thais before, that was also a very pleasant joint work, and we can also see that there are a lot of complementarities between what we do. So that's also very nice, and it's been great to develop and sustain also those this collaboration work. Now, of course, there are some people also in the group that I would like to thank, in particular Adeline, because it's we've been working together for a few years now, and that was one of the initiatives that we could really strengthen this cooperation within our ESD group. I've really appreciated that, and also all the input from Adeline, especially about biochemistry, bioinformatics. I've also learned a lot from her, so that's really wonderful. And now, Adilson, you also mentioned Veronique and Joel. Well, they really helped a lot. Thank you very much to you for the support also in the lab for all this extensive purification that has been performed. And then there are many more people actually in the group who have been involved, because I think it's one of the characteristics of your project, Adilson, that it's been touching upon many aspects of what we can do in the group, with the colleagues working on lipids, working on anti-nutritional factors, working on fluorescence spectroscopy. So, many people have also helped, and that's also really great to see this initiative of really gathering a lot of people and a lot of expertise in the group. So, I'm not going to mention everyone. That would be a lot of people, but thank you very much to all. And now, of course, you, Adilson, it's been really a great pleasure to have you around in the group for 1 year. It really went very fast, but you really became a fully integrated member of the group, interacting with all those people. Very organized, because it has been said, you really did a lot of work in this short period of time. And I think it's also really thanks to your great organization skills. Always in a good mood. I was also very impressed by your very positive, eager, and very calm also attitude. So that's at least apparently. I don't know how it was inside there sometimes, but that's really a great pleasure to work with you, Adilson. And also I've been really impressed by your capacity to really pick up the things that we said in meetings and also really use that in the best possible way to also make progress with your research. So that's really precious that you have it, and I really I'm sure that you will also make the best use of it in the future. And I really wish you all the best for your next personal and professional project. So. Thank you very much. And now I give the floor back to you, Mr. President.
Thank you very much.
Thank you very much, Professor Bertin. I will now ask Mrs. Véronique Solé-Jameau, who is an invited member on the board, to please share her comments.
Yeah, thank you so much to invite me to be here in the committee. It's a real pleasure for me, and it says a lot for our work. And I wanted to thank you.
I think the microphone is not working.
Yeah? Yeah? Okay, thank you. You can hear me, Adilson? Yeah? Okay. So it was a real pleasure to work with you.
Your microphone is off. No?
Okay.
Okay. No.
I think Pedro is having an issue with hearing us.
Yeah.
You can hear me?
Yeah.
Yes.
Yes.
I can hear everybody but Véronique.
No, I cannot hear Véronique.
Can you speak, Véronique? Let us.
Yeah. Can you hear me? Everybody is okay? Okay.
Yeah.
Not problem.
Otherwise, Véronique, you just run to the closest office and you can speak from there.
Yeah, but I just finished and I come to your desk. Yeah. Sorry, Pedro. Just I wanted to say that it was a real pleasure, and I learned a lot about Brazilian and Brazilian way of life with you, and it was very nice to work with the Brazilian clan with you and Thais. And my last word for you is obrigado. So I come to your desk, Claire. No? Okay. It's okay for me, I finish.
Okay, thank you very much, Mr. Now I'll request Dr. Adeline Bois, who was also a co-supervisor, to please share her appreciation with us.
Thank you very much, Mr. President. So I'm the last one. The afternoon is ending. So I would like to join Claire and Vania to thank the committee members. It was a very, very interesting afternoon, and I really appreciate the questions. It was a bit hard to find to fill the, let's say, the guidelines for the jury with the co-tutel, but I'm very glad because we went to very let's say, process engineering aspects to very specific biochemical questions. So it was very rich, and I have many notes. So for the future, it's interesting. So of course, thank you, Adilson. It was a really great pleasure to work with you. I'm very impressed by your ability to work hard, even in an environment that is very different from Brazil. So here you arrived. You had different skills, different background, but you managed in 1 year to learn a lot and to do a lot. So it was, very nice. And I have to say that you're a student that is very easy to supervise because you're very well structured. Your experiments are very well performed. Guillaume talked about the very small standard deviation on the forming properties.
So I think I'm not hearing Adeline.
You don't hear?
Me neither.
I cannot hear either. And I cannot hear Véronique, actually.
Yes,
Yeah.
for me.
I was perfect.
Because in the YouTube, it seems that I am seeing Adeline, but here in the...
They are with the microphones on. I don't know what is happening.
Yeah.
Okay. Are you hearing Adeline properly, Pedro?
No.
No one is
Me
really.
neither.
Okay.
Okay.
No one is hearing me, right?
You can hear Adeline to
She should
in
maybe
the
get out
YouTube.
and in back
Yeah.
to the meeting.
Maybe Pedro, you can log out and logging
I'll
in.
go out and return. Let me see.
I'll do the same.
So let's the people to log out and to come back. Adilson, you were able to hear everybody, huh? Okay. Pedro, is it better now?
Could you speak something, Adeline?
Yes.
Yes.
Okay.
Oh, yeah. I'm sorry.
Let's wait for
Unlikely things. Yeah.
But I think there was a global problem because
Okay.
so now everyone is listening is hearing me.
Are we missing somebody?
Now perfectly. Yeah.
Okay, good. So
We are missing somebody. Remy is okay. Remy is back. Okay, very good. Okay, please resume.
okay, so it will be short. First, thank you to all the members of the committee. It was very rich afternoon. I really appreciated all the questions and the discussion, and so it's also a good way to celebrate Adilson's work by interacting with him and discussing on the scientific matter. Yeah, I can go directly to you, Adilson. Thank you very much for all your hard work. It was a real pleasure to work with you for a year and beyond, because you went back to Brazil like 6 months ago, and we have been still working across continents. So I'm very impressed. I was saying, but I feel like I'm repeating myself, but you didn't hear it, so it's good. I was very impressed by your work ability. Like in 1 year, it's really impressive how much work you have done. Everything in a very calm mood, as Claire said, like very calm, but step after step, you managed to do everything. And the jury, they highlighted your writing skills, and again, it was very easy to supervise your work, because you were very good in writing, very good in communicating your results. The infography are always very clean, very easy to understand. So yeah, so for us it was kind of an easy task to help you in your way of your PhD, and I really hope that you manage to continue, maybe in academia or in private sector, but you're a very good scientist from what we've seen, and yeah, I wish you a really great success. I hope we keep in touch, and maybe in the future we work again together. We don't know, but at least good luck. And and also because to thank Claire and Vanya. It was very, very nice working with you in a very nice environment, and I learned a lot for the past years, and yes, maybe we'll work again together. So thank you, President, and the floor is yours.
Okay, so thank you very much to everybody for all these very, very interesting discussions and questions with Adilson. I think now we will go into a private session for discussion between us, and we will invite people who are not within the discussion to leave temporarily this session, so that we can proceed with the deliberation on our side. Okay?
I will stop the transmission, and then we will back. Okay?
Okay. Yeah.
Yes, we have to wait some seconds because there is a delay in the transmission. Just a minute.
Okay.
Adilson. welcome. Adilson, can you see if it's the streaming is now?
Yes, I will check here. No,
it's not easy, this life of broadcasting. I think it's okay. It's transmitting. Yes.
Yes.
We can go on.
Okay.
Okay. So, Mr. Adilson Roberto Locali Pereira, after careful consideration of your manuscript and after hearing you defending your PhD thesis in front of your jury today, and on behalf of all the members of your jury, we are happy to say that you are admitted to the diploma and the grade of PhD in Nantes University in France and in Sao Paulo State University in Brazil. Congratulations. Okay.
Thank you very much.
And in the specialty.
Thank you, everyone. I can speak a few words, or we need to?
Yes. Before you speak a few words, we will request, as per required by our university in France, that you make an oath. Okay. So you will need to now to address to all your peers that you will now based on your work for the future on some ethical issues, aspects.
Okay, perfect. Here, can I start? Oh, sorry.
You can read it, don't share.
Yes. Well, in the presence of my peers, with the completion of my doctorate in food science, in my quest of knowledge, I have carried out the many research demonstrating intellectual rigor, ethical reflection, and respect for the principles of research integrity. As a pursuit for my professional career, whatever my chosen field, I pledge to the greatest of my ability to continue to maintain integrity in my relationship to knowledge, in my methods, and my results.
Very good. Okay, congratulations again, Adilson.
Thank you very much. I would like to thank all the committee members for the presence, Thank you very much, everyone. Well, thanks for the nice talk that we had. It was very enriched to discuss the project. I think that we can see the project in different ways when we do things like that in the defense. And also, I'd like to thank my advisors, so Vania and Claire and Adeline, for all the knowledge that was shared, and for being very patient with my learning process. And well, with everything, you were very open to give new ideas, and you were very dedicated to supervise my project. So thank you very, very much. And thank you also to all the people from INRAE that helped with different parts of the project. It was a lot of people, and they contribute a lot. And to you all that are watching, thank you very much. And to my family and my friends. And that's it.
Thank you. Again, congratulations.
Thank you. Thank you.
Congratulations.
Thank you.
That was very nice. So now we call you Dr. Adilson.
Yeah. Now I am relaxed. At the beginning, I was very anxious and nervous about it. I think it will take some time to absorb the new information. Yeah.
Okay.
Thank you all.
Yeah, so that's a little bit the weird part of this video conference setup, that now we have just to say goodbye. But well, I'm sure that we will keep in touch, Adilson, no doubt about this. And I also look forward very much to have a chance to see all of you, the committee members, in person, hopefully soon. And well, people at ESD for sure, we will also no doubt about this. That's right. Thank you all for a great morning, afternoon. Well done, Adilson. Yeah, and well, we speak very shortly, I'm sure.
Sure. Okay, great. Bye-bye, everybody.
Bye-bye.
Thank you.
Thank you very much.
Et voilà. And bye, Vanya.
Thank you very much for everything. I am a very shy to say, but in these occasions, but thank you very, very much.
And thank you. To be honest, it was a really, really great pleasure. And I was a bit sad when I heard that you were already retiring, because I was seeing like a great collaboration.
But we can manage some things to do. Okay, we can talk.
No, but I mean, but thank you so much for your kindness. I really felt that you are a very kind person, and it was really great to work with you.
You too. Thank you. Bye.
Bye, Vanya.